So thank you all for coming. It's been a wonderful career in aging. And I just wanted to start off by talking about the fact that I've been almost indecently fascinated by islands, I think probably from the time I was a boy when I read Robert Louis Stevenson's Treasure Island. And as a consequence, a lot of the work that I've done in aging has started from the fact that I thought you could learn a great deal from islands. And here's a map of the tropical Pacific showing just some of the islands that I've worked on there. I also worked on some islands in the Atlantic Ocean. But I want to use this as a lead-in to talk about a very specific population on a very specific island. Before I do that, I didn't know how much of a lay audience this was going to be. So I just wanted to describe what I mean when I talk about comparative biology of aging. And that's just that one can learn something by informative comparisons among species, which is the way that many people think of comparative biology. But also that one can learn something by informative comparisons among groups within a species. And I like to think that one of the things that I've done a few times in my life is sort of point out what's obvious. That is, point out what everybody knows, but very few people really see or think about. And what I'd like to start with today is what strikes me as a major facet of the biology of human aging that we really don't consider. And we don't consider the implications, and we don't consider what we know in detail, and that's the biology of sex differences in aging. So I'll start off here with a story about an island, the island of Iceland, and I'm starting with Iceland because it's probably the place in the world that we know the most about the human biology of. Iceland, as many of you know, was settled in the late 9th, early 10th century. We even know the names of many of the original settlers. We know a great deal about its history through the years, and its history has been described as a millennium of misery. It's on the edge of how humans can survive. This is a landscape from Iceland, and over the course of the 1,000 years that people have inhabited Iceland, it's been beset by an enormous number of catastrophes, famines, floods, volcanic eruptions. Every time that happened, the population of the island would crash. So surprisingly, the population of Iceland in 1400 was just about the same size as the population of Iceland in 1900. Now, as a consequence of this huge series of catastrophes, the other thing about Iceland that makes it a geneticist's dream is that because there's a relatively homogeneous population, like I said, they know the names of many of the original settlers. It's really ripe for genetic analysis, and in fact, there's a company that's been trying for years to take advantage of that genetic analysis. Now, because of Iceland's history, it's had these enormous rises and plunges in life expectancy over the time. And so I'm just going to focus on when the really good demography started, which was the early part of the 19th century. And if you look at life expectancy, the upper two lines there are the male and female life expectancies at birth, and the lower two are life expectancies at age 50. And you can see that over the course of the 19th century, life expectancy dropped as low as 17 or 18 years and went as high as nearly 60 years. But despite that, and despite the different causes for those different accelerations and decelerations, one thing that was continuous throughout this time is that women's life expectancy always exceeded men's. And in fact, if I just put up there the percentage of the difference in life expectancy of women's and men over that century and a half now, you can see that it's always above zero. So when times were good, women live longer. When times were bad, women live longer. It's absolutely striking to me. Now, Iceland has been protected for the most part from wars, so that's not been a part of this problem. The other thing to note is that this is not unusual. Women live longer everywhere. Here are 143 entities from the UN Demographic Data Book, in comparing the percent superiority of female life expectancy over male, and you can see that 140 of those, there's a clear female advantage. There's one that's 1-to-1, but this is not to be taken seriously. This is Afghanistan, and this has got exactly the same life expectancy with no decimal point for both sexes. I think they've given up trying to do censuses in Afghanistan. And the two lower points there are highly questionable. They're small countries that, to believe the data, you'd have to believe that they have a higher male life expectancy by a considerable amount than the Japanese, which are typically the longest lived group in the world. So in good times, in bad times, in Western countries, in Eastern countries, in Southern countries, in Northern countries, women live longer. It's also true that they survive better at every age. This again is Japan. This is the demography of Japan. I could have taken it from virtually any country. This is the annual probability of death as a function of age, and you can see that right there throughout the life course from birth. In fact, women survive better than men even before birth. The sex ratio early in conception tends to be about 1.5 to 2 males per female, but males die at a higher rate, so it's by birth it's about 105 males to females. So whatever is causing this difference in survival begins to operate very early in the room and continues to very late in life. And I'd like to use the comparative biology today to really ask 3 questions, and typically not to answer any of the questions, but to ask them. The first question is, why this sex difference in longevity that's so robust? I think it could be called one of the most robust aspects of human aging that we know about. What's responsible for that? Now, when I bring this up to lay people, they come up with a couple of answers right off the bat. The one you might call the cognitive disablement hypothesis. And this is that males do stuff that's so incredibly stupid that, of course, they would survive at a lower rate. And while that may be true, it's hard to attribute it to things in the womb. Another hypothesis is the hypothesis of testosterone dementia, that males have this instinctive recklessness. It's either that or it's heart disease. I said, well, everybody knows that men tend to die of heart disease earlier, but it turns out that all these things may play a role, but it's more robust than that even. Here are the top 12 causes of death in the U.S. as of 2006. And you can see all the bars that are above the line mean that men die at higher rates of those. And you can see that for 10 of the 12, men die at a substantially higher rate. Stroke is about 1-to-1. The one disease that tends to be harder on women than men in terms of mortality is Alzheimer's disease. So this superiority of female survival cuts across cultures, cuts across historical epochs, cuts across ages, and cuts across causes of death. So the question becomes, now if men lived as long as women, if men survived as long as women, they would get a bigger longevity boost than if we cured cancer. So this is a very, very substantial difference. It seems to me that it's a difference that we should know more about. Because although there are a few general hypotheses, none of them is very satisfactory. So how would we go about investigating this? Well, biologists would like to get in, tear things apart and tinker, and humans typically would object to being castrated early to see what the effects of that is, or all being fed the same diet or given the same stress. So the first thing that we would do, of course, is that we would go to one of our experimental animals to ask the experimental questions of what's the mechanistic basis of this difference in survival. Now, an interesting thing about laboratory mice is that if you go to the literature and ask the question, do male or female mice live longer? That is, what kind of model might these be of this longevity superiority? The answer you get is yes. That is, you can find statements in the literature. Of course, everyone knows that male mice live longer than females. And other statements the same year, everybody knows that female mice live longer than males, and other people that say that everybody knows that there's no difference in longevity in mice. And it turns out they're all right. I went to the literature and came up with about 90 studies of survival studies of mice and asked the question, did the males or the females live longer? And you can see that there's a pretty nice even distribution. And the first thing you think, for those of you in the audience who aren't biologists, a lot of the mice that we study are highly inbred and genetically identical to one another. And there are various strains of these, and there's one strain, the C57 black 6, that for reasons that I don't understand has kind of become the industry standard, not just for aging research, but for most biomedical research. So one explanation potentially for this variation in male versus female longevity is that maybe it's just these different inbred strains. But I found about an additional 25 studies that were all of the same inbred strain. And those are the open circles there, and you can see even for the C57 black 6 mouse, you see this enormous range of variability, and it's something that we have no idea about what the cause is. If we knew something about the cause, if we could replicate the cause, it probably has to do, most of these are in very similar mouse colonies. They're in pathogen-free mouse colonies, they're eating similar diets. If we could understand and predict when males would live longer and when females would live longer, then maybe we could make some headway towards understanding this mystery. But I haven't been able to figure out anything. And the other possibility, of course, is to expand the research bestiary. And for those of you who know me, you know I've been harping on this for years, that we really need to take the 10 million species of animals out there and investigate more than 3 or 4 of them. And so on this panel here, we've got animals that males live longer, animals that females live longer, and animals of which there's no different species. So there are possibilities for investigating this. Now, what I often hear is, well, isn't this pattern of enhanced female longevity a general pattern in nature? And the answer is, I don't think so. It's easy to find species in which males live longer, and as this shows, females live longer. It's also easy to find species in which you get the other. So if we expand the bestiary, it may be that we can make some headway into understanding this longevity difference, which is if we understood it and we really knew how to replicate it, we could enhance the longevity of men by more than if we cured cancer. Now, that's only part of the sex difference in aging mystery, however. The other part of it is that a lot of the treatments, now we've been wildly successful over the last 15 years, I'd say, in finding genetic approaches and pharmacological approaches, and then we've known for a very long time about nutritional approaches that make animals live longer. But one of the things that we've tend to ignore was, in a lot of these approaches, sex matters. So here's one study of a genetically modified mouse that, if you read the literature, what it says is that this genetic modification makes the mice live longer. And it's true for the females. For the males, it looks in this graph as if they're slightly longer lived, but in fact, that was not a statistically significant difference. And there's a study about to be published of the same mutation in which, again, the females live longer, although it's a smaller amount, but the males clearly do not. So at least in this one case, there's a genetic mutation, a genetic treatment. In this case, it's not a mutation, it's a direct deletion. There's a genetic treatment that has a major effect on females and not an effect on males. Now, one of the most robust findings associated with changes of longevity is that animals that live longer tend to be more resistant to a broad variety of stresses. And if you take these same mice and you ask them how well do they survive in response to an oxidative stress, you find the same thing that you find in longevity, that there's a big effect on the females, but not much effect on the males. Now this hardly seems fair. If females are already living longer, at least in humans, and now they're also responding better to genetic enhancement of longevity. However, it's not always the female advantage. Here's another one again. You read in the literature, this is a recent paper from Science, a very nice study, that knocking out this particular gene makes mice live longer. Well, in fact, it does make mice live longer as long as they're female mice. If they're male mice, it doesn't. So understanding these sex differences in longevity, I think, is going to be important to understanding not just sort of naturally occurring differences, but also therapeutically occurring differences. If we look at some life-extending pharmacological treatments, here is a couple of studies from the intervention testing program. In both of these studies, there's a small but a very real and a highly statistically significant increase, in this case, for males in response to these pharmacological treatments. For the same drugs, there's no difference whatsoever in females. In one of the cases, we kind of understand why that is, and the other one, we don't. So these sex differences in aging are robust. They're important in even the animals that there doesn't seem to be a consistent difference in survival. Now, the second question that I think this comparative biology of sex differences can give us some insight on is the relationship between lifespan and health span. Now, for many years, those of us in the field constantly harped on the fact that what we're trying to do is make people live longer. We're using animals to try to make people understand how to make people live longer. And one of the things that happened in the early 2000s when I got to testify in front of the President's Council on Bioethics is that for the first time I was challenged. I always thought this was the greatest thing since sliced bread. And everybody would agree that it's the greatest thing since sliced bread. But I found while being interrogated by the people on that panel that not everybody thought it was the greatest thing since sliced bread. And subsequently, when I gave public talks, I would often say, I have a pill here. It doesn't cost more than an aspirin, and it makes you live 50% longer. How many of you people want to take the pill? And usually most of the people would raise their hand. They would want to take the pill. And then I would say, okay, now should we give this pill out to everybody? And then nobody would raise their hand. So there's this suspicion, this worry about increasing lifespan. But you ask the same people, how would you like to increase your health? How would you like to preserve your health? How would you like to have the health at 80 that you had at 50? Then you get a whole different series of answers. So it gradually dawned on those of us in the biology of aging community that it was really health span that people were interested in. I mean, we knew this all along. It was our shorthand way of talking about it, and we often assumed it was the same thing. So a few years ago, the NIA had a summit. The National Institute on Aging had a summit where they sort of planned, where should we go from here? One of the things that came out of that summit, thanks to a number of the people in the room here, is that we really should focus on health span rather than lifespan, making the assumption that they may not be exactly the same thing. And I think this sex difference just points out exactly what we mean and why we have to be serious about that. Because as robust as the long life of women relative to men is, equally robust is the fact that women are generally less healthy than men throughout life. This graph here shows, for a variety of Western European countries, the percentage of women of 50 plus that have some sort of functional, some sort of physical functional problems. And you can see a couple of things in that. Very worrisome from the standpoint of the US, I would say, is that the fraction of people with functional problems is higher in the US than any of those other countries. The other thing, though, is that women in all of those countries have more functional problems than men. And this is something, again, that everyone knows but not everyone appreciates. In fact, women have more physical illness over the course of a lifetime, more days in which they're disabled, more hospital visits, more hospital stays, more doctor visits. And this isn't only true of Western Europe. This is also true in places like Bangladesh. So an equally robust pattern for human longevity is that despite their greater survivalship, women have poorer health. And if we could understand that difference, and we could keep women alive as long as they are now, but we could increase their health, then we'd be doing a great service to humanity. And as a consequence of that, there's been a renewed emphasis in the field on understanding health span. And various places, including San Antonio, have begun to develop what I call mouse health span clinics, where we measure their body composition, their muscle strength, their agility, their gait. Well, we have lots of fancy stuff, and we're beginning to put this together. And I think over the next few years, we're going to start to understand when lifespan and health span converge and when they don't. So the third question that I think we can learn from the comparative biology of aging is, again, what can we learn from animals that age successfully? And for those of you in the audience who aren't biologists, you may not realize this, but all of the traditional animal species that we use in aging research, we use because they're demonstrably unsuccessful at aging. They die quickly, they fall apart in a hurry. It makes them experimentally tractable, and it's wonderful. And over the last 15 or 20 years, we've made enormous progress in understanding the genetics and physiology of aging by studying these animals that are demonstrably unsuccessful at it. What we hope and what we assume is that if we learn how to make a mouse that normally lives 2 years live 4 years, that will give us some insight into how to improve and extend human health as well. But it's entirely possible that there's a complementary approach. And that complementary approach is to do what the pharmaceutical companies have been doing for years. So this is a flower called the rosy periwinkle, and from it is produced a potent chemotherapeutic drug for cancer called vincristine. And the pharmaceutical companies have taken advantage of what Leslie Orville says, that evolution is smarter than you are, and have gone out prospecting in the natural world for things that are good at treating diseases, in this case, for things that are good at inhibiting cell division. And it's been wildly successful. I mean, most of our pharmacopoeia comes from natural products, at least originally. We may improve on those. So the idea is that nature has found ways of combating the destructive processes of aging as well that are better than we have. So in this graph, which I stole the idea from George Martin, shows that researchers have really been able to increase the life of a mouse, let's say, from 2.5 years to 4 years, but evolution has changed the mammal from 1 year to over 200 years. So evolution has clearly come up with some tricks that researchers not yet have done. So as a consequence, I've come up with basically a list of candidate species that I think might be particularly interested for aging, that I've been calling recently Methuselah Zoo, after the biblical patriarch that lived 969 years. I guess one of the things would be to try to figure out how Methuselah lived 969 years, but that's going to be difficult. Now, studying these exceptionally long-lived species has some challenges and has some complications. One of the complications is that you'll notice these species are very, very different in size, and one of the things we know, almost as robust as the sex difference in survival, is that size counts when it comes to mammals. That bigger mammals live longer than smaller mammals, something all of us intuitively know. A mouse is shorter lived than a dog is shorter lived than a horse. And so it's important to figure out whether there might be small animals that live a very long time for their body size that might be able to teach us something about aging, rather than looking at animals that only live an absolutely long time. Because small mammals and birds are very different. They have exponentially higher or faster just about everything that you can imagine, from metabolic rate to heart rate. The old idea that I spent 20 years trying to kill is that everything in aging is determined by metabolic rate. And so this idea that the number of heartbeats per lifetime is constant among mammals is something that you hear again and again and again. It's one of these ideas that's so intuitively pleasing that you just can't seem to get rid of it. But there's something there. There's something there, because having all the processes go faster in a small animal presumably would make it live faster in some elemental sense. And to try to standardize this in some sort of simple quantitative way, my graduate student, Kate Fisher, and I years ago developed something we call the longevity quotient, which is just a way to measure how long a mammal, in this case, lives relative to how long you'd expect it to live for its body size. So let's take a hypothetical animal there with a circle around it. That's how long it actually does live. That's how long it should live, given that body size. And so the longevity quotient is simply the ratio of that. And it's important to note that that's a logarithmic scale on the y-axis there. So that is way, way above. So if you actually retool our bestiary, and now you start thinking about it in terms of longevity quotient rather than absolute longevity, you can see that there's quite a wide range. The mouse and the rat, the laboratory mouse and the rat, it's quite obvious why we use them now. They're extremely short-lived for their body size, which again makes them useful in the laboratory, but has some disadvantages as well. The other thing to note about that is that humans are not the longest-lived mammal, either absolutely or relatively. That there are animals such as Brandt's bat that live virtually twice as long as they do. So, let's just talk about a few of these animals. Species with high LQs are the ones that I think we ought to focus a lot of energy. There's an arrow at where humans are. There's an arrow at where one of the most interesting of these species is the naked mole rat. The naked mole rat has recently become a celebrity organism in the biology of aging because although it's the size of a mouse, it lives about 10 times as long as a mouse. Rochelle Buffenstein at my institution has been studying these for years, has really been instrumental in getting them involved into the aging bestiary. And just recently, there was a very, very nice genome sequence of the naked mole rat published in Nature. Like I say, it's not that there's one naked mole rat that lived to be 30 years old. They live to be old often and routinely. So this is the kind of thing that we can learn a lot about. My personal favorite for Methuselah's mammals is the Brandt's bat. This is a bat that's this big. It's 7 grams. It lives more than 40 years in the wild. And I've got in the wild italicized for a reason. It has a longevity quotient of almost 10. The reason is that any animal in the laboratory does not have to really maintain a high level of physical fitness in order to survive. But any animal in the wild does. So to point out a few things about bats, bat longevity records are virtually all accidentally acquired. Nobody's going to start out to study longevity in an animal that lives 40 years. I wouldn't have even tried that when I was a graduate student. So most of what we know about the bat longevity is accidentally acquired. It may be considerably underestimated. The other thing is that because these things survive this long in nature, they must be able to face and overcome severe challenges. For instance, we're not the only ones that can figure out when millions of bats pour out of a cave opening every night. Predators can do that, so bats have to be able to avoid that. They hunt with high-frequency hearing. They yell and they listen for the echo. And the first thing to go in human sensory biology, of course, is high-frequency hearing. So they can do that actually better and for longer than we can. They need to keep spatial memory. They fly 50 or 100 miles a night. They must find their roost again by morning. So let me finish up by one other brief discussion of a way that you can make surprising insights, potentially, from animals that you would never expect. And that is to use comparative biology to address some human aging problems. Now, there are a variety of neurodegenerative diseases that in the past have been called protein aggregation diseases because a hallmark of the disease has been some cluster of insoluble proteins, and these are examples of 3 of those diseases here. Interestingly, a new hypothesis is that not those protein aggregates themselves that cause the problem, but that those are a consequence of smaller aggregates, ones that are still soluble, and that actually when you get the aggregates and they fall out of solution, that's actually a protective mechanism. And so we may be in the middle of a paradigm shift in terms of understanding these protein misfolding diseases, which might be a better way to go about it. Which brings me back full circle to Iceland. Because off the coast of Iceland in 2007 was discovered what I'll call Methuselah's mollusk. One of the things about bivalve mollusks, which is the kind that have 2 shells that you can open and that many of you will probably eat for dinner tonight, is that every individual out there in nature is carrying its birthday on its back. If you section them and you do all kinds of fancy cutting and polishing and etching, then what will be revealed are growth rings so that you can tell the exact age of every individual in nature. And when you do that, you find some remarkable things. Here's a freshwater oyster that lives as long as 200 years in nature. And here's Methuselah's mollusk, the ocean quahog, Arctica Icelandica, because the longest one was discovered off Iceland, that lives over 500 years in nature. Now, we tend to think of mollusks as it's kind of a lump of flesh, but in reality, they've got a complicated anatomy and physiology just like we do, including a beating heart. So we have out there in nature these animals that have hearts that have been beating since before Shakespeare was born. And it's possible that we can learn something important from understanding things with such extraordinary longevity. And so let me just describe very briefly a project that we've been doing. My collaborators are listed at the bottom there, and the person who did the work that I'm going to briefly mention here is in capital letters, Steve Treaster. What we've done is we've taken a series of these bivalve mollusks, because not all of them are long-lived. Yes, the longest lived ones lived over 500 years, but there are also species that only live a year or 2. And the idea is that by looking at the biochemistry of these, we might be able to understand something about the mechanism. And I told you before that one of the most robust findings we've discovered over the last decade or so about longevity is it tends to be linked to stress resistance. And here's an indication of the stress resistance of a handful of mollusks that we actually did in the Woods Hole course that Gary Ruvkin and I direct one year. And what is shown here is that the longest lived clam indeed survives the best under this kind of oxidative stress that we used. In fact, there was a very clear dose-response curve. Now what we did, we've been looking at proteins and how proteins aggregate in these various species, and one of the things that we discovered was that if we looked at the fraction of these aggregated proteins, the non-aggregated proteins, when the animals were not challenged, when they were comfortably in their normal habitat, there was a nice decline. So animals that aren't stressed, these animals that aren't stressed on a species level, the longer lived they are, the fewer aggregated proteins they have. But when you stress them, when you put them under some sort of stress, you get an absolute reversal of that protein aggregation, suggesting that there's an active process by which the long-lived ones can take damaged proteins and aggregate them and have them fall out of solution. We think it quite likely that the longest lived mollusks seem to have some sort of active aggregases, which may have, in fact, be protective against misfolded proteins. An implication of this is that an animal that doesn't have a brain might actually give us some insight into neurodegenerative diseases. So let me just finish with the following. I think I try to make a case that sex differences in aging, health, and longevity we really don't understand at all, But it's worth investigating in detail. Not only do we not understand them, but there's virtually no research in this area. Novel research may hold keys to understanding not only sex differences in aging, but also may provide understanding of how to prevent or treat specific human diseases that we never suspected in the past. So let me just finish with two things. First is a thanks to the people who gave me this award, and really the thanks is owed to all of my friends and colleagues and sponsors over the years, and you know who you are. Many of you are in this room. And I'd also like to thank my parents, because as you can see my father here with the red circle around his face. My father grew up as a migrant farm worker, and nobody in his family had ever gone to college. Very few of them had actually gone to high school. But together, they ensured that I became the first person in my family on either side to go to college. And here I am. So thank you very much.
Don't go.
Not so bad.
Okay, questions?
Steve, the principles of animal longevity. The principles of animal longevity, as you sketch them out, apply to plant growth.
That's a very good question. So can you use animal longevity trends to understand plant longevity? I have a tough time conceptualizing plant longevity because they're modular organisms. You know, you pull a piece out of a plant and you stick it in the ground and it grows a new plant. And that doesn't happen when you pluck an arm off. So there's a different way of thinking about plants. Now, I have friends in the evolutionary biology community that this drives absolutely nuts. They say, no, understanding plant biology of aging is exactly the same. But to me, there's a big conceptual difference there. And I don't know how to think about it. I wish somebody would help me learn how to think about it. But you know, you can have an aspen clone that's a hectare in size, that's 10,000-year-old, but they could all be disconnected at the bottom. So it's almost like, you know, every few years you plucked off your thumb and you threw it someplace and it grew a new human being. How would you think about aging in that case? And I just don't know how to do it. Yeah, Dick and George.
Just a follow-up on what Tuck just raised. Are there any animals with death hormones that would...
With death hormones?
Which you get in plants?
Well, I would say that Pacific salmon probably have death hormones. They get this huge burst of glucocorticoids, which basically causes an immune system collapse. There are a few, but they're not a lot. They're actually, one of the interesting things about plants is the ones that only survive a year are pretty common. Whereas in animals, we know of some animals that have this very abrupt death, but doesn't seem to be nearly as common. So that's another kind of mystery. And maybe those can teach us something different than something like a bristlecoat pine or an aspen clone or something. George, did you?
Steve, so how do you feel about how far you can go on the sex differences on the notion of heterogametic versus homogamic sexes? So you know, the female has an advantage of somatic mosaic, so it buffers recessive change.
Right.
And the quail male lives longer as heterogamy.
Yeah.
What do you think about that?
So that's probably the most intuitively satisfying idea. The idea is that we have these two sex chromosomes in mammals. It's an X and a Y, and you only have one copy of most of the genes on the X chromosome if you're a male, and that sort of exposes any detrimental genetic effects there. And in things like birds, you have the reverse, where it's the female that has the two different sex chromosomes. And there is a general pattern for males to be longer-lived in birds and females to be shorter-lived in mammals. Maybe. I'd say there are more species in that direction than the opposite, but the problem is that's a pretty universal trait, but the sex difference is not universal. There are mammals in which males are longer lived, and there are birds in which females are longer lived. So that is a problem for the big universal. Yes, in the very back.
So Steve, can't the sex differences that you see be explained by reproductive strategies, and also the differences that you see in small animals that live a very long period of time? Take the brown bat, for example. It lives 20, 40 years, long-lived small animal. But its reproductive strategy is very, very, slow. It reproduces once a year or so. It has to live longer in order for the species to survive, just in the same way as a mouse that lays down a lot of offspring very quickly. It doesn't need to live quite as long.
Well, I mean, there's clearly a tie-in between reproductive strategies and longevity. In fact, evolution is working on the reproductive strategies. The longevity is a kind of secondary consequence of that. So clearly that plays a role. And in understanding sex differences, ultimately, I'm sure sex has an impact. But if you actually look at the data, I mean, there are these anecdotal data out there. What happens if men are castrated or women are ovariectomized? And it's not highly uniform like you might expect. But this is the kind of thing that you really could look at in experimental animals and hasn't really been done extensively. It's actually been some very nice work by Jim Carey recently in mouse on this. Thanks, Greg. Aubrey?
Yeah, so another question about the sex differences. So you very clearly emphasise that it's not just a species thing, it's not even just a strain thing, but even the same strain, you have this enormous variation in what you see. I mean, what that tells me is that there's a lot that we don't know about how to do the experiment in a reproducible way. And one thing that I've been banging on about for several years now is the fact that the husbandry of mice, even though we think we know how to do that, seems to be an enormous variable in terms of longevity. The colony that Tom Kirkwood had at the University of Newcastle is well known by those of us who come to conferences to live a full 6 months longer than their average C57 black 6 colony. That's 2 whole mortality rate doubling times. So that means a factor of 4 difference in age-specific mortality. And nobody has the faintest idea why. I'm wondering whether this is something that has not been nearly prioritized enough in what we do.
Yeah, I would agree with you. I would agree with you, Aubrey. There are subtleties of husbandry that must be behind this, and there's a lot to learn. I absolutely agree. Yes.
I commiserate with Tom for his extra expenses. The question of protein aggregation.
Yeah.
And I mean, I think it's a little bit sexy, but it's also a little bit dangerous. Clearly, the first option is to degrade a damaged or an unfolded protein. So you've got a balance here between proteasome and non-protease degrading these damaged proteins before they can aggregate. And then you've got a short period where you've got a solubilized aggregate versus an insolubilized aggregate. And of course, the aggregates will inhibit the proteasome too. That's been demonstrated many times over. So I'm a little nervous that the experimental condition determines the outcome rather than the biology determining the outcome.
Well, I would agree. I mean, this is not a fully developed story yet, and I would not make the claim that the protein aggregates are not the problem. But I do think it's an intriguing new way of thinking about protein aggregates. Yeah, Tom.
I just wanted to thank Kelvin for his concerns about the animal husbandry costs, but there is a very clear benefit, and I think the husbandry is important. Anecdotally, we had an experience where we were asked to supply some mice from our colony to a research group at another university that had been sourcing their animals from a commercial supplier. And after 6 months, they got back to us and said, it's very strange, but the age-related change we were studying previously does not appear in your animals. So what I think they were studying was not aging, but a readout of chronic stress. So there is an important message in terms of the quality and the longevity of animals. But that brings me on to the question that I really wanted to ask you. I totally buy the case for looking at some of the longer-lived animal models that you were advocating. But those are going to be very long studies. And I think what one needs to do then is to try to devise a system that really can accommodate longer-term studies in species like naked mole rats or, or the bats, because we have a research machine at the moment operating that does not easily accommodate studies in organisms with lifespans much longer than 2 to 3 years.
So you're proposing a 20-year postdoc?
Well, I mean, I'm not proposing. I'd like Steve to answer the question.
But I guess you put me on the horns of the limit here, because there's part of my research that I'm advocating. It's really nice to have these animals that are shorter lived than any of these other. But I completely agree that to study aging, it would help to have a longer grant window, as one might say. However, about these extremely long-lived animals, to try to understand how an animal lives 500 years compared to another animal that lives 10 years, you don't have to study them for 500 years. You know that difference exists. The idea is just to try to figure out why it exists. But your more general point, yeah, if I only had to apply for a grant every 10 years, it would be wonderful. Yeah, if I leave it in. Okay, Jim.
So in our fruit fly studies, over 20 years we've studied these both sexes, and we've come to the conclusion that everything is context specific. You cannot make the statement one sex lives longer than the other. And so even with humans, in the 2,000 males in the 2,000 environment live longer than 1,900 females. So I mean, how would this idea that you're trying to identify all these different species, which lives longer than male and female? I think it's context specific.
Well, I would say the mouse data suggests that it is context specific. However, that's very hard to reconcile with the human data that is so robust, because in the same environment, there's a very, very robust sex difference in humans. I have to admit that how robust that is in other species, we don't really know about at this point. But I think the mouse data and your fruit fly data fit together very, very nicely. That the only difference is we don't understand what the conditions are in the mice. Yeah, Gary.
These sorts of gender differences, intersexual individuals, can be informative. And I don't know if you have enough demographic sort of oomph on this, but people who seek sex change operations, is there any, if you can look at any point in their life and ask actuarially, what's the death rates?
That's an interesting challenge for the demographers in the room, I would say. And there are plenty of them. Yeah. Don?
Provocative. Thanks very much. Two questions on the gender issue. First, I know it's been done, I just can't call my memory. You look at the prehistoric record. There have been good studies of that, for prehistoric cultures, even real or even exist today, in terms of the gender difference.
Yeah, I've reviewed that. The prehistoric data aren't good because the ways for aging skeletons are not good. But then you have the modern hunter-gatherers that you can assume are something like prehistoric. And the data are highly variable. There's a number of difficulties with it. It's usually small sample sizes, usually exact birth dates. But you can find even several different studies of the same group, because the differences are small. If you figure that the differences are, let's say, 5%, but life expectancy is 20 years, then even a 5% difference is less than a year. So that would take some very sophisticated demography with some very reasonable sample sizes. And so I don't feel confident that we can say anything about that.
Second question related to gender difference. Bruce Kern's presentations weren't very intriguing. It just has to address this issue of women having a mortality, maybe having a morbidity advantage, as you pointed out. Is there a twist on antagonistic biotrophy here? When maybe it's a benefit in some aspects and not others? What's going on?
So in terms of how that fits with that intent, usually the first person to jump at any explanation that involves antagonistic pleiotropy, but I don't see an obvious connection there. Is there a question, George?
Steve, when we're phenotyping mice from a cardiovascular point of view, if they can make it, the females can make it through pregnancy and support a pregnancy, they typically have a pretty good cardiovascular system. How do you reconcile the fact that I've never had a baby and my cardiovascular system has never been so challenged? There's really a selective genetic pressure, isn't it? Or is it all the same?
That's an interesting idea. Virtually all the studies that I showed were mice that had never been reproductive. But you might argue then, but the capacity is there, and therefore, you know, but if that's the case, you'd expect there to be a generalized sort of female advantage, and at least in mice, I think it's pretty clear that there isn't any big generalized. Yes?
You may have some comment on this. Demographers seem to write as if they know the answer to the cause of human difference in sex difference and life expectancy, making positive statements about the relative role of genetic environmental causes. The thing is really, as you said, quite a mystery. It was only until rather recently that for all the countries, life expectancy of females was higher than males. For many years, perhaps for centuries, in several countries in Southeast Asia and Central Asia, Iran, Pakistan, etc., male life expectancy is higher. Now today, the range is terrific, you know, from the Soviet Union with the difference of 10 and down to just a few years in some of these same countries that had the male excess. The variability is perplexing. The United States had just a difference of a few years back in the early 20th century, perhaps because of the excess mortality in childhood. But then it grew rapidly upward, up to 8, and now it's back down to 5. It's really quite a mystery as to why this is, although, as I say, demographers have been able clearly to point to environmental factors in this historic change, which couldn't be genetic, obviously, the one I just mentioned.
Yeah, it is interesting. One of the things I've noticed over the years is that the UN Demographic Data Book typically has a country or two that has greater male longevity, but never persists. Those countries, the next survey, have flipped around. Not very much, but they have flipped around, which I think suggests that there really is some sort of robust biological difference. If the differences were always switching back and forth, I'd almost be willing to say there's very subtle environmental or cultural things. But this just seems to be consistent enough to really get my attention.
Can I ask you a short other question? This is a mystery among demographers. And you mentioned that the primary sex ratio of deaths at conception was like 150. Of course, sex determination is not possible at that age of life. Now sex determination is possible perhaps for 4 months or so. But the studies that have been done usually have related to late fetal life where clearly male mortality is higher. How did you decide just extrapolating backward?
No, there are good studies from very early, from 16 weeks in gestation, but they're done cytogenetically. They're not done anatomically. So cytogenetically, it's not hard to tell males from females, right after conception.
It used to be said, and I don't know if it was actually true then and whether it's true now, that humans are the only mammals where bred females live longer than virgins, and that no other mammalian species is that true. Is that actually the case? And it would be for social support reasons, obviously.
There's actually some pretty interesting recent information from David Waters in dogs that females that are intact the longer they're intact, the longer alive they are relative to females that aren't. So I'm always suspicious of things that start humans are the only species. I'm just intuitively suspicious of those things, and so I'm equally suspicious of that one. Serge? Yeah.
Is it possible that the high morbidity in women is because of high mortality in men? Because weaker individuals would be just eliminated by high mortality, so the only ones that last are sort of stronger organisms, that's why men have less.
But remember, it was a higher mortality from prenatally.
Right, so that is why the morbidity of women is higher, just because men sort of
Oh, so that's the explanation for the difference in morbidity. Reasonable idea, Ron?
So with the mollusks, you have the opportunity to take on whatever is the favorite cause of aging. So there's stem cells, inflammation, etc. Do you have time and/or resources to live?
Yeah, there are a number of other people that are. I don't have the time or the expertise to do all that myself, but there are other people that are. For instance, Irina Comboy is very interested in the muscle stem cells in the clamps, and so I'm hoping that we get a constellation of people who are interested in exploring their favorite hypothesis.
Surprisingly, I have a follow-up question to what Ron just asked. So you showed two parameters which have a perfect correlation between the lifespan of the lamb and the plants that you measured. I'm sure you have a few which there's not. Therefore, that the hypothesis that that's the meaning of the medical question. Have you had results as well that you did not show?
So did we always get this? So here's, this is a really important point of philosophical. Let's say you do the same experiment 10 times, and 6 times you get one result. Is that what you mean?
You tested the hypothesis that aggregates were
right,
and you got the perfect correlation. You tested the hypothesis that susceptibility to say the stress was important.
Right.
You tested the hypothesis that DNA damage is important and you did not get there.
No. So we haven't eliminated.
Tests that have been positive. Wow.
Right. Yeah. Ultimately, you would like to know that. We are currently in a phase of the primacy of proteins rather than the primacy of the image. Okay. Well, thank you very much for coming. A geneticist's dream is that because there's a relatively homogeneous population, like I said, they know the names of many of the original settlers. It's really ripe for genetic analysis, and in fact, there's a company that's been trying for years to take advantage of that genetic analysis. Now, because of Iceland's history, it's had these enormous rises and plunges in life expectancy over the time. And so I'm just going to focus on when the really good demography started, which was the early part of the 19th century. And if you look at life expectancy, the upper 2 lines there are the male and female life expectancies at birth, and the lower 2 are life expectancies at age 50. And you can see that over the course of the 19th century, life expectancy dropped as low as 17 or 18 years and went as high as nearly 60 years. But despite that, and despite the different causes for those different accelerations and decelerations, one thing that was continuous throughout this time is that women's life expectancy always exceeded men's. And in fact, if I just put up there the percentage of the difference in life expectancy of women's and men's over that century and a half now, you can see that it's always above 0. So when times were good, women live longer. When times were bad, women live longer. It's absolutely striking between time. Now, Iceland has been protected for the most part from wars, so that's not been a part of this problem. The other thing to note is that this is not unusual. Women live longer everywhere. Here are 143 entities from the UN Demographic Data Book in comparing the percent superiority of female life expectancy over male. And you can see that 140 of those, there's a clear female advantage. There's one that's 1-to-1, but this is not to be taken seriously. This is Afghanistan, and this has got exactly the same life expectancy with no decimal point for both sexes. I think they've given up trying to do censuses in Afghanistan. And the two lower points there are highly questionable. They're small countries that to believe the data, you'd have to believe that they have a higher male life expectancy by a considerable amount than the Japanese, which are typically the longest lived group in the world. So in good times, in bad times, in Western countries, in Eastern countries, in Southern countries, in Northern countries, women live longer. It's also true that they survive better at every age. This again is Japan. This is the demography of Japan. I could have taken it from virtually any country. This is the annual probability of death as a function of age, and you can see that right there throughout the life course from birth. In fact, women survive better than men even before birth. The sex ratio early in conception tends to be about 1.5 to 2 males per female, but males die at a higher rate, so it's by birth it's about 105 males to females. So whatever is causing this difference in survival begins to operate very early in the room and continues to very late in life. And I'd like to use the comparative biology today to really ask 3 questions, and typically not to answer any of the questions, but to ask them. The first question is, why this sex difference in longevity that's so robust? I think it could be called one of the most robust aspects of human aging that we know of. What's responsible for that? Now, when I bring this up to lay people, they come up with a couple of answers right off the bat. The one you might call the cognitive disablement hypothesis. And this is that males do stuff that's so incredibly stupid that, of course, they would survive at a lower rate. And while that may be true, it's hard to attribute it to things in the womb. Another hypothesis is the hypothesis of testosterone dementia, that males have this instinctive recklessness. It's either that or it's heart disease. I said, well, everybody knows that men tend to die of heart disease earlier, but it turns out that all these things may play a role, but it's more robust than that even. Here are the top 12 causes that everybody knows, but very few people really see or think about. And what I'd like to start with today is what strikes me as a major facet of the biology of human aging that we really don't consider. And we don't consider the implications, and we don't consider what we know in detail. And that's the biology of sex differences in aging. So I'll start off here with a story about an island, the island of Iceland. And I'm starting with Iceland because it's probably the place in the world that we know the most about the human biology of. Iceland, as many of you know, was settled in the late 9th, early 10th century. We even know the names of many of the original settlers. We know a great deal about its history through the years, and its history has been described as a millennium of misery. It's on the edge of how humans can survive. This is a landscape from Iceland. And over the course of the 1,000 years that people have inhabited Iceland, it's been beset by an enormous number of catastrophes. Famines, floods, volcanic eruptions. Every time that happened, the population of the island would crash. So surprisingly, the population of Iceland in 1400 was just about the same size as the population of Iceland in 1900. Now, as a consequence of this huge series of catastrophes, the other thing about Iceland that makes it So thank you all for coming. It's been a wonderful career in aging. And I just wanted to start off by talking about the fact that I've been almost indecently fascinated by islands, I think probably from the time I was a boy when I read Robert Louis Stevenson's Treasure Island. And as a consequence, a lot of the work that I've done in aging has started from the fact that I thought you could learn a great deal from islands. And here's a map of the tropical Pacific showing just some of the islands that I've worked on there. I also worked on some islands in the Atlantic Ocean. But I want to use this as a lead-in to the talk about a very specific population on a very specific island. Before I do that, I didn't know how much of a lay audience this was going to be. So I just wanted to describe what I mean when I talk about comparative biology of aging. And that's just that one can learn something by informative comparisons among species, which is the way that many people think of comparative biology. But also that one can learn something by informative comparisons among groups within a species. And I like to think that one of the things that I've done a few times in my life is sort of point out what's obvious, that is, point out